Hello, good afternoon everybody. It's Thursday the 9th of April now, and it's the last day before Easter, so welcome to the last day this week of lockdown bioinformatics. So today we're going to finish off our first kind of bioinformatics exercise, proper bioinformatics exercise on the course, which we've done this week, building on our previous lessons to at the command line, working with standard Linux tools. We've written a script this week that will download some data from Uniprot, and we've also looked at installing the software that we need and then running the software that we need to build a multiple sequence alignment from that downloaded data. And today, a piece of software which is specifically designed for working with viewing, editing multiple sequence alignments, and that's a piece of software called Jalview. So a couple of orders of business first to deal with before we crack on with what we're going to do today. The first of which is that when we looked at the sequences that we downloaded yesterday, and we looked particularly at the final sequence, which looked from the sequences that we downloaded like it was a lot, for one thing, was a lot shorter than all of the other sequences. All of the other ACE sequences that we downloaded didn't look quite right. Also, the thing that I particularly noticed about it, given that I picked these 12 sequences to download, was that I didn't really recognise the organism that it came from. Now, I had intended to include, as the kind of 12th sequence in the alignment, one which was a bit less similar to the bulk of the... So I kind of worked my way down. I did a blast with, just to kind of give you the background to how I generated these. I did a blast search on Uniprot with the human ACE2 protein sequence. And then I kind of worked my way down the blast results and got homologs of ACE2 with kind of varying levels of similarity to the human protein. So beginning with, I think the first one, other than the human one, is the gorilla ACE2 gene, which is obviously going to be very similar in terms of its protein sequence. I think it's something like 99.9% the same as the protein, as the human protein, down through other kind of vertebrate mammals and out towards some more kind of outgroup ones. Like, I think we looked yesterday, one of the ones that we saw, it looked like it might have been anaconda. I've actually looked it up. It's American chameleon, not anaconda, which is a little bit of a shame. It would have been quite opposite if it was anaconda. Anyway, the 12th one that I wanted to add was the kind of most remote relative that it still made sense to include in a multiple sequence alignment. So I didn't want to be going out to kind of 20% identity. I wanted to keep things relatively similar. So I picked a homolog from a species which I knew, which was Xenopus, and it was the thing that was clear to me when we looked at those sequences yesterday, kind of previewed them before and after we built the alignment, was that final sequence wasn't from Xenopus, and it obviously wasn't labelled as being an angiotensin converting enzyme either, which is what ACE stands for. So I was a little bit concerned that actually I'd had an error in transcribing the Uniprot identifier when I wrote down my list. Maybe we were downloading the wrong protein, and I've done a little bit of work this afternoon to figure this out, and yes, I was right. We did indeed download the wrong sequence. I'm not entirely sure what happened when I wrote down the ID. The ID for the Xenopus ACE protein begins with the same 6 characters as the ID for the protein that we downloaded. So I got the last 4 wrong, but they're really very wrong. It's not that I've missed one character or slightly mistranscribed. They really are quite different. So the first thing we're going to do today, and this is a good opportunity to kind of just run back over what we did yesterday is I'm going to change that last identifier to make sure that we download the right protein so that our alignment makes a little bit more sense, because aligning, it can be useful to have an outgroup in your alignment. So it can be useful to have one protein which kind of isn't that similar to all of the others. Otherwise, your consensus can be really quite, your alignment, if your alignment is kind of too, the proteins are all too close to one another, then there's not much information in the alignment. Because things haven't diverged too much, so you're not seeing the really conserved positions in the alignment. So I wanted a bit of an outgroup, and that can make a lot of sense. But having your outgroup as a protein which is completely unrelated to all of your other proteins in your alignment really doesn't help matters at all. So we're going to just go back over what we did yesterday, and while doing so we're going to change that identifier so that we download the right protein. So that's the first thing that we're going to do this afternoon. Okay, so we need our VM. So let's pull up our VM. Here we are, and Control L just to clear my screen, and then I'm going to list my present working directory, and I'm going to remove both the input and the output for our script. So I'm going to remove my muscle input and my muscle output. Okay, so RM, we can do this just with muscle underscore and then a star, and that will remove just those two files because they're the only two files in this directory which contain the text muscle, begin with the text muscle. So that's my RM. That gets rid of the input and the output files, and we can recreate those using the script that we finished writing yesterday. And then we want to edit. First of all, we want to edit our Uniprot IDs file, and this is where having your kind of input data stored separately from your script can be really useful, and also having your input data in one place. The tendency when you first start to write scripts is that you might type in your IDs by hand into your script, and if you end up using them more than once in that script, then you're going to type them in more than once, and that is error prone, but also means you have to find all of those places in order to edit your Uniprot IDs or whatever it is that's your input data. In this case, our input data is separate from our script, so it gives us one place to make that change to change this incorrect ID for the correct one. So I'm going to use nano to edit my Uniprot IDs.txt file. Then we're going to go down to the bottom line, and we're going to change this. So if you recall a couple of minutes ago, I said that the actual protein that we want, the identifier starts with the same first 6 characters. So A0, A1, L8, they're unchanged, but it's not E5, F5, it is HCX9. Okay, so that is the Xenopus laevis angiotensin converting enzyme protein. So that's the protein that we actually want to download, as opposed to the one that we were downloading yesterday, which wasn't the right one. So we'll save this, Control O to write to a file, then Control X to exit. And then we're going to edit our script because now we want to re-download that data and reconstruct our alignment. So we're just going to edit the script to make sure that we know exactly what the script is doing. Okay, so nano get Uniprot data. Okay, so here's our script. So the first thing that we do at the top of the script is we activate the appropriate conda environment. And I'm doing this within the script because this means that if I run my script, if I forget to change my environment when I log in and I run my script from my base environment, for example, this first line will make sure that the right conda environment gets activated. And so we're definitely going to have the software available to us to actually construct the multiple sequence alignment. So our muscle aligner, which is installed in this MSA environment, is going to be available within the script because we've activated the environment within the script. So that's an important step to make sure that when we come back to this script again in the future, we can make use of it. We can actually we don't need to remember what the right conda environment was that we used when we ran this script. Okay, then the second line of code, which is actually about the fifth line of the script, takes in our Uniprot IDs. So this is the thing that is reading that input file and giving us a variable in our script, this VAR variable within our script, which contains those Uniprot identifiers. So now that we've changed that Uniprot IDs file to contain the correct identifier for our 12th protein, this will now read in those specific identifiers. And the change that we've made in that file will be reflected when this script runs this time round. Okay, so then we have a second variable, which is the stub for our Uniprot URLs for the Uniprot API, and then we've got our for loop, which, if you recall, the for loop now downloads all of the data. So for each of our Uniprot IDs, it goes to the Uniprot API and downloads the FASTA formatted sequence for that protein. It then adds the contents of that file that's just been downloaded into our muscle input file. So we're using standard output redirection with the double greater than, which means append to the file rather than overwrite the file, to create our muscle input file with the contents of all 12 of our protein sequence files that we've downloaded from Uniprot. And then because we've taken a copy of it into that input file, we can remove the file that we've downloaded from Uniprot, because we don't need to keep that, as well as our copy of the data that we've put into our muscle input file. So that this loop goes around 12 times for each of our 12 Uniprot identifiers, and then when that's finished, we can actually build the multiple sequence alignment using muscle. So that's what this last line does, is actually runs muscle to make our multiple sequence alignment, which we make in this output file, muscle_output.clw. So that's our output file. So because at the very top of the script we activated the right conda environment, muscle should be available in our path because we've installed it using conda. And so this should run fairly smoothly, and I haven't made any changes, so I'm just going to Control X to exit, and then I'm going to execute that script. So we point to where the script lives, which is in our present working directory, and then the name of the script, get_uniprot_data.sh, to execute it. And this will go and download those 12 files. We've seen this enough times now. It was interesting. We've seen this enough times now that we know what's going to happen. So it downloads those 12 files and builds our alignment. So we've run our muscle alignment here, and so now we've got in our present working directory, after running that script, we again have the muscle input.fasta file, which we'll just take a quick look at. Okay, so here we go. We've got our ACE proteins from our various organisms. So Homo sapiens, gorilla, pongo, macaque. I don't know what Calithrix jacus is. Felis catus is the cat. Then you've got rat and mouse. Something else I'm afraid I don't know. The Anolis carulensis, that's the American chameleon. Danny O'Reilly, so that's the zebrafish, and then this is the change that we made. This final protein that we've downloaded now is quite similar in length to all of the others. The header for this particular gene now tells us it's an angiotensin converting enzyme, so it is an ACE, and it's from Xenopus laevis, which is what I expected. So we have now downloaded the appropriate protein, and that will also be reflected in our alignment. So our alignment now should be slightly more cohesive. So rather than having one sequence which has a huge number of gaps in and doesn't really align very well at all, like we did yesterday, we should now have a much more cohesive alignment where things make a lot more sense. So the Xenopus gene, which is the second one down in our alignment, does actually look a reasonable amount like all of the other ACE genes, ACE proteins in our alignment. So it does now look like we've got a decent set of relatively related proteins, and that our multiple sequence alignment now is actually reflecting much more the conservation across this protein family. So where we have conserved positions, which are indicated by the stars at the bottom of the column, those conserved positions are now going to be much more informative as to the biology and the biochemistry of the ACE gene family, which is nice. That's better than our position that we were in at this time yesterday. So I know it's taken the best part of 20 minutes to get back to the point that we left it yesterday, but I think one thing, repetition is always quite useful, but also we've actually managed, we have corrected a mistake that we made, and I will kind of point out, and I know this is a point that maybe I'll belabor as we proceed, but we discovered that mistake, and it was a mistake. You know, I will quite happily hold my hands up and say that I when I wrote down the identifier, I wrote down the wrong thing. I'm not entirely sure how I did it, but that's what happened. But we discovered the mistake, and we did it because we were examining the inputs and outputs for our commands. We were looking with a critical eye at the files that we were writing to our disk. That when I looked at the sequences that we downloaded yesterday, and when I looked at the alignment that we constructed yesterday, with a critical eye, we could see that it wasn't quite right. It wasn't, the thing that we'd produced wasn't really quite what we expected. And particularly, one of those proteins didn't seem to be annotated as being an ACE protein. It had a different name. And the other thing for me was that it came from an unexpected organism. So by looking at the inputs and the outputs from our various commands with a mind on the fact that humans make mistakes, we made sure that we didn't take that incorrect data from the input from the beginning of our process all the way through to the end. And that's a relatively instructive thing to have done, and I am not going to claim that I did that on purpose. Although it is quite useful sometimes to make these mistakes on purpose. That isn't what I did here. Believe me, this was a genuine and honest mistake. But hey, we found it, and that's I think a positive thing to take forward from today. So we've corrected our mistake. We've built our alignment, and actually this clustal format for a multiple sequence alignment is quite nice to look at. It's okay. We can look at it and we can see the conserved positions and the similar positions because they're marked in the alignment, and it's written in such a way that it kind of makes sense to the eye to to scan down this alignment and see where you've got like big gaps, for instance. So in this particular ACE gene here, we've got what's clearly a big insertion compared to the other members of the family. So there's a stretch there of 30 or so amino acids that none of the others have. And it's flanked by an extremely well conserved region, both to the N-terminal and C-terminal ends of that insertion. You've got an extremely well conserved chunk of amino acids, which is potentially quite interesting. Okay, so and this cluster alignment lets us see some of these features and lets us see where we've got perhaps a really well conserved domain in the protein, where you've got no variation at all among these really quite diverse species in the sequence of this protein. But we could do better than this. It's a nice way of looking at the alignment, but it's not perfect, and there are things that we can do better than this. So what I wanted to do today was to take a look at a way that we can do better, and we're going to do that using a piece of software developed at the University of Dundee called Jalview. So Jalview has been around for certainly as long as I've been in bioinformatics, so 15 years or so, probably longer. I'm not entirely sure of when it was first developed. And it's a really, really good quality and robust tool for working with multiple sequence alignments, particularly multiple protein sequence alignments. It's a really nice piece of software for doing that. And it's not a command line tool. So unlike I think pretty much everything else that we've used so far, it's not a tool that you can work with at the command line, but you can launch it from the command line. So we can run it as a command from the command line, and it will open us a graphical user interface containing Jalview. And importantly, we can also install it from the command line. And that's the thing, is at the moment we haven't got Jalview installed on our system. And we haven't necessarily got the dependencies for Jalview either. It's a Java application, and I don't think Java comes installed on Xubuntu by default. So we'll also be missing dependencies. And what do we do about dependency resolution and installing software? We look at conda. And the nice thing, which I have discovered in the last 24 hours, is that Jalview is distributed via conda these days, which is great. So if we Google Jalview conda, then you will see that Jalview is available to us in Bioconda, and we can install it via the Bioconda channel using conda, which is great. That's the system that we've set up. That's why we've set this system up, is to give us access to this kind of software. So we're going to use conda to install Jalview. We've already made the Bioconda channel available to us in conda. We've updated our conda configuration. We did this on Tuesday when we installed conda. We've updated our conda configuration to make the Bioconda channel available, so we don't need to explicitly name it when we run the install. So to install Jalview, the command is conda install Jalview, which is fairly straightforward. So this will resolve all the dependencies that Jalview needs to make sure that it can run properly, and it will install Jalview, and it will give us an executable in our path. So now hopefully conda will go away, and we'll within our MSA environment, we'll figure out what it needs to install Jalview, and we'll resolve that environment, and we'll install it. And hey presto, there you go. And what you will see is, whereas muscle had very few dependencies, Jalview has quite a lot of dependencies. So there's quite a few things that we need on the system for Jalview to work correctly. And this is where a system like conda is worth its weight in gold, because these are things we would have to either the Jalview installer would have to install and we wouldn't entirely know what it was doing, or we would have to satisfy some of these dependencies ourselves before installing it. So, for instance, one of the things it's going to install is OpenJDK. That's Java, so that's the Java programming language. So JDK stands for Java Development Kit. So one of the things that we're going to have to install to be able to run Jalview is an installation of Java. We would otherwise have to do that ourselves because you wouldn't be able to install Jalview without it. So Conda is going to resolve all of this for us and do it in a fairly transparent way. It's telling us what it's going to install. So off we go. We're going to proceed with that, and it's going to download all of those different applications and get them installed and get us up and running so that we can run Jalview. Okay. So this will take a few. I'm just looking at the comments and seeing this thing about conda init has. I noticed that when I ran my script as well, the conda init hasn't been activated. I think it might be. I think it might be because we're running it in a naive shell, because we're effectively running it with bash, so it's not running within an environment which conda has been activated at all. And so I think it might be relying on your path to be right, and it's not really. Sorry, I'm thinking aloud. So it might be that conda activate MSA within the script doesn't work at all for various reasons. So I think the suggestion that Peter has there, which is to point to an activate script to do it, is probably the right way to do it. But yeah, that's an interesting one. I hadn't really thought about that, but I did note when we executed the script before, I did notice that a very similar warning to that conda init hasn't been activated warning. So I think maybe having the conda activate within the script we've not done it in quite the right way, and we might need to go back and think about that another time, maybe for a future script. Okay, so clearly this is taking a while to download because some of these pieces of software are quite big packages. So Jalview was quite big, OpenJDK is pretty big, and on a domestic broadband connection, these things just take a little while. So I'm going to have to do the live streaming equivalent of mugging to the camera while this is downloading. I think by the time we've got to the end of today, hopefully we'll have achieved a decent kind of little mini bioinformatics project this week, which is quite satisfying for me. I've quite enjoyed doing it. I think when we come back after the Easter weekend, after the long weekend, we will begin to turn our minds to our goal of analysing some RNA-Seq data. So I think what we'll do next week is I'll locate an appropriate experiment, and I've seen a couple of candidates, so I'll pin down an appropriate experiment that we can think about analysing, and we will figure out next week how to get the data, which might be via we might be able to use wget if it's available over FTP, or it might be that we need to figure out SRA tools if we have to download it from the sequence read archive directly, because that can be problematic. So that's going to be one exercise, and then we need to start to think about how we're going to analyse it. So we'll need some software at the command line to do our initial kind of QC and quantification analysis, and I'll do I'll do an introduction to RNA-Seq just so we're all on the same page about what we're actually trying to achieve with RNA-Seq analysis. And I think that will probably take most of next week, if not all of next week. And then the week after that, we will we'll turn our attention towards R, and I'll do some introduction. I'll do a similar introduction to R that I did with the command line. So a nice, slow, from the beginning introduction to the R programming language, the R statistical programming environment. And we will start to build up our skills in R with a goal of analysing that data that we will download next week. So I think that's the plan for the next. I'm going to guess that's going to take at least as long as we've spent so far. So I think that's probably going to be the next maybe 3, maybe 4 weeks of work. Okay, so conda has finished. So conda's downloaded all of my dependencies, so it's downloaded all of my dependencies and installed them, and I should now have in my path a Jalview executable. So there you go. I typed JAL and pressed tab, and it auto-completed to Jalview. So I now have a Jalview executable that's been installed by conda. I've never tried to launch Jalview at the command line before, so I don't know what the options for Jalview and the arguments for Jalview are to get it to work. So let's see if there's a help file. Let's run Jalview-h and see if we can get some help. And as I said, I've not done this before, so this is as new to me as it is to you. I have used Jalview before, but not for a while, and I've not launched it from the command line before. So here's some options. Okay, so there's some command line options. Our usage, there you go, is Jalview open file. And I think that's probably all we need, to be honest. There's a lot of options, but they're all about creating alignment files, creating image files. So I think that's for our output. But just to run Jalview with an alignment file will be Jalview-open file. Okay, so let's see if it works. Jalview open, and our file is our muscle output file. So the cluster W formatted alignment that we created in our script that we ran earlier. So Jalview open muscle output.clw. And off we go. So Jalview is going to try and open. Okay, some errors going on in the background. I'm not going to worry too much about those because Jalview has opened in the foreground. So you can see our terminal is being very, very noisy in the background. But Jalview has popped up in the foreground. So this is Jalview. And Jalview has opened. And I'm just going to say, do we want to collect usage statistics? Sure, why not? Let's close the news and let's maximize Jalview. Maximize. Right, okay, so it has summarily failed to read our input for some reason that I don't entirely understand, but this is clearly not our alignment. So the first thing that I'm going to do is I'm going to close that alignment because that hasn't worked, and then I'm going to try again to input an alignment from file. So Jalview is a graphical user interface, so we should be able to use it just as a GUI. Lots of point and clicks. So let's find the file that we want to open, and it is it's a cluster W. So Jalview wants our cluster file to be called .aln. Okay, so let's rename it. So let's go back to our terminal. So let's have a new terminal. File, open tab. There we go. So new terminal. Because Jalview is running, it's taken this terminal over. So it's showing. It's just showing the output, the standard output and standard error that Jalview produces as we use it. So we can't run commands in this terminal at the moment until we close Jalview. So I've opened a new terminal. So I did that in the file menu, and I picked open tab. So I've opened a new terminal in another tab in the same window. So that's my clean terminal. So my conda environment here is base. Because it's a new completely fresh terminal, which would be opened in my home directory. This terminal is open to be in the same working directory as my other terminal was open. Okay. All of that said, Jalview is expecting our cluster alignment file to be named .aln, not .clw, which we named it yesterday. So I'm going to move my output file. So I'm going to move muscle output from muscle_output.clw to muscle_output.aln. Okay, so that it obeys the conventions that Jalview expects. Okay. So, okay, list. So there we go. That's changed the name of my output file. I'm also, while I'm at it, I'm just going to edit my script just to, so that the next time I run if I run this script again, it will produce the output file with the quote-unquote right name. So I'm just going to change that to .aln as well. There we go. So that next time I don't need to worry about moving the file around. Hello, somebody flagging me in the comments. Can't open Jalview on Windows. Okay, so that is potentially because... So conda doesn't distribute a Windows version of Jalview, I don't think. We don't have to run Jalview from within our VM or from our command line. We can run the web start version if we want, if we've got Java installed on our machine. So let me just quickly show you. I need a browser. And I need to go to jalview.org. Okay, so this is on my Mac. This is not on my virtual machine. This is on my host machine. It used to be, okay, so you can use WebStart to access old versions. So we can install Jalview locally. And because we're using the share on our VM, or if you're running all of these commands locally, you'll have your files on your local machine. You can install Jalview without using conda, and certainly I can do that on my Mac. So I could download a Jalview installer. There's a Mac OS version. There's the instructions for installing via conda, which is tested on OS X and Linux. There's almost if you go here on a Windows machine, there will be a Windows installer here as well. So I can download the installer and then install it on my host machine, not on my local machine. So I'm going to download it and install it. Improvising slightly. That'll take a little while because it's quite big. Okay, it's going to take a minute or so to download. And then once that's downloaded, I can run that installer and get things running locally rather than relying on, and get things like a standard software install rather than necessarily relying on conda to do this for me. There is other ways and means of getting this running. There is also kind of web starty versions. So we could run old versions via Java Web Start, where you don't need to download the application itself. There's also, in theory, Jalview in your browser as well via Jalview JavaScript. How well this works, I don't really know, but it is in theory possible to run it in your browser as well. I've downloaded the installer for macOS, which I will just quickly run. So there you go. Jalview to drag into my applications directory, which hopefully will do something. Doesn't seem to be doing anything. Okay. So open a new Finder window. Applications. Jalview. There we go. 4th of July 2019. I wonder if that's an old version. So there you go. Here's my applications directory. I'm just going to drop Jalview in here and replace the existing version. Okay, so there's Jalview. So I'm going to double click. There we go. Open it, and here it is starting up. So this is me running it locally I've not installed via Conda. I've installed via the installer that they distribute on their website, and Jalview is going to run for me relatively happily on my local machine. So here's Jalview starting up with all of its all of the kind of gubbins that it contains. So when it starts, normally you would see all of this, all of these example files that it loads up for you. So let's close those and let's open. So input alignment from file, and I'm going to load. So I'm going to go to my share directory, which on my local machine is VM files, or was it lockdown? It was lockdown learning, wasn't it? There you go. So this is my share directory, but this time on my Mac, not on my VM. Go to week 3 and go to muscle output.aln. That's my cluster format output from my muscle alignment. I'm going to click open, and that has also failed. I wonder what's going on here. Okay, I'm going to do something else as well. So clearly it doesn't think that this is actually a cluster alignment output, and I don't really know why that is. I suspect it's because of this header here. It thinks that it's reading the alignment where it isn't actually doing that. So muscle presumably hasn't produced something which is strictly speaking cluster output. So what I'm just going to do, I need to activate my conda activate MSA, and then so I've activated my MSA conda environment, and I'm going to run muscle at the command line with the same input, muscle in, muscle input, sorry, .fasta, and I'm going to produce the default output of muscle, which is a fasta formatted alignment, because I know that this will be in the right format. So muscle output this time, but instead of being it being a cluster output, it's a fasta output, so we'll call it a fasta file because that's what it is. I'm sorry about the messing about here, but as I said yesterday, I don't really get time to test these things in advance, and I think it's relatively instructive for you to watch me debugging things as well, because it's kind of showing you the real process rather than you getting a very sanitized, or here is A, B, C, and D without any kind of faffing around. So now I've got a second output file. This time it's fasta formatted. rather than being cluster formatted, and hopefully that will load more sense. It's less human readable, so we can have a look at it, and you'll see that it's less human readable because each sequence is kind of shown in isolation. So this sequence and all of its gaps and everything is shown completely dissociated from all of the other sequences. But it's hopefully a bit more machine readable, and Jalview will be able to understand it better. So let's load it in two places. Let's load it here. So what format do we want? We want FastA format. Missed by one. 010. There you go. So muscle output.fastA. Okay, so that's our FastA outputted alignment from muscle that I just produced at the command line. Again, I can go back in and change my script to make sure that this is what's coming out from my script as well, and then click open, and that's more like it. So now we've got an alignment. So if I maximize that window, I'll do the same. I'm going to do the same in my Mac version as well. So here's Jalview, which I downloaded and installed on my Mac. So we go File, Input Alignment from File, and it's muscle output.fasta is our alignment, and it's FastA formatted alignment. There we go. Why are you not working? To open. That's interesting. Why are they grayed out? There we go, it's worked this time. Don't know why it didn't work last time. But there you go, there's again is the, so it's the same thing. That's the alignment opened in my directly on my Mac in the version of Jalview that that I downloaded on my Mac and installed locally. Because I share the files between the VM and the Mac, I can see the files without any other kind of messing about. So if I want to be able to work with these files in my local operating system, because of the way that we set up the VM right at the beginning, we're able to do that. So now we can view this alignment and we can see some of the features of Jalview that make it really useful for viewing the alignment. So as well as kind of an overview of the alignment up at the top here, which is even more human readable than the cluster output because it's all on one. There's no kind of separate blocks of the alignment separated out with new lines and things. Instead, we've got the alignment all in one long line, and we can scroll along it by going left to right, and we can look all along the alignment this way. As well as that, we also get some overview tracks here generated by Jalview. So a score for the conservation of the alignment at each position. So how conserved is our alignment at this position, and where we have positions which are absolutely conserved. So for instance, this leucine here at position 16 in the alignment, this is absolutely conserved. So we have a really high conservation score. It's actually got a star because it's completely conserved. The alignment has a measure of quality, so how good is the alignment at this point? That's to do with the gaps that have been introduced and how well conserved your alignment is kind of across the block and things like that. You've got a consensus sequence along with how much of the alignment is represented by that consensus. So here you've got methionine, which is present in 92% of the positions in the alignment, and that's because there's one of our sequences which has a serine there, not a methionine, and so on. So those are really quite useful kind of overviews of our alignment, and we can kind of scan across the alignment and by looking at those these sort of conservation consensus tracks, we can identify blocks of the alignment where you have really strongly conserved regions between all of these species that we're working with here. So you can see those regions where you get this really kind of a run of conservation. We can also colour the alignment in, so we can apply colorings based on various different colouring schemes. The clustral colouring scheme is usually quite a good one, which again adds information to your alignment, which allows you to see things at a glance and see where there are deviations in your alignment, where there's conservation and what sort of conservation it is. So residues are getting coloured in according to their properties, and they're coloured in most of the time only if they're present in most of the rows of the alignment. So this is again quite useful information that Jalview can add into our view of the alignment to make this a more useful way of viewing the alignment and understanding a little bit about what's going on. There's that gap that we talked about, that insertion in one of our species, where we've got this kind of a sort of 30 to 40 amino acid insertion, which is flanked by these very highly conserved regions that we could see in the cluster output, and we can also see here. We can identify really quickly here in the Jalview output. And there are other stretches of really strong conservation, like this big long chunk here as well, that again we can identify really readily in this view of the alignment. So this is kind of really neat and a useful piece of software just for viewing the alignment, but also we can also do other things with Jalview. Jalview offers us other functionality beyond just viewing an alignment. So we can, from this alignment, we can use Jalview to construct some sort of kind of tree of the relationships between these sequences. So we can use Jalview to give us more information than just the alignment. We can use the alignment to calculate, say, a neighbour joining tree, for instance. Although I don't quite know why this is all greyed out. Maybe we have to highlight all of the sequences. Yes, there we go. You had to highlight the sequences. So highlight the sequences, and then that allows you to calculate a neighbour joining or an average distance tree from that alignment based on a particular substitution matrix. So we're going to use the Blossom 62 matrix because actually it makes a lot of sense for the relationship between these sequences. Press calculate, and we get a tree which is not a very interesting tree because these are all really quite similar proteins. They're all members of the same family. There's not a great deal of kind of deviation between them, so we get a tree that's really quite boring. It also might be that this is based on what's highlighted, and it may be that actually what's highlighted is a single column or a small range of columns in the alignment. So let's just check by doing select all and then recalculating our tree. That's better. So I think I probably selected a single position in the alignment. So now the tree's a little bit more informative, and shows us the relationships between our ACE sequences, and that they are, to be honest, they're kind of what we would expect. The Xenophus sequence, which we have now included successfully today, and was included as an outgroup, is indeed an outgroup in the alignment. The non-mammalian species here are also kind of more distantly related from the core of the alignment, and these are all of our mammals here. So we've got the human and the gorilla sequence really quite closely related, as we would expect. So it's quite useful. Jalview is quite good at going from a multiple sequence alignment to these kind of quite useful visualizations and kind of analyses of those alignments. There's other functionality that we could explore with Jalview, but we're kind of running out of time, and I think it might be instructive for you to kind of, now we've got this alignment and we've got it loaded up in Jalview, to sort of take some time with that alignment yourselves and with Jalview, and learn a bit about Jalview, have a look at the online documentation for it, and figure out what you can do with Jalview as a piece of software. It's really got a large amount of functionality, so it's really worth taking some time to explore it if this is the sort of thing that you're interested in. Okay, so everything that I've done there I've done on my Mac, but I could equally have done in my virtual machine as well. And so we could, if I just pop back to my virtual machine and close this Jalview instance, back to my terminal, do Jalview open. It was muscle output.fasta is the one that works. So this will, there we go, open Jalview for me from the command line on my VM, and it opens the alignment. So we've started with the alignment open. And again, if we select all, calculate tree, there you go, let's calculate the neighbour joining tree like we did before. Same tree comes out. Whoops. Neighbour joining tree, there we go. So you can see that the version on Linux in our VM is working in exactly the same way as the version that I've got installed on my Mac. Admittedly, in the VM, that's all quite tiny, so maybe you can't see it quite as well as you can see it on my Mac. This, being able to install software on the VM and run it is really, really useful. But also being able to get to the files on your VM on your local machine and install software on your local machine if you need to is also a really useful thing to be able to do. And that's part of the reason why I set things up the way that I did back at the beginning, 2.5 weeks ago. Okay, so that's it for today, and that's it for this week, because we have a long weekend. So happy Easter to everybody, and I hope that you all have a restful break this weekend if you possibly can. I will return on Tuesday at 4 p.m. For our next week of Lockdown Bioinformatics. As I mentioned while Conda was doing its thing earlier on, we will turn our attention to some RNA-Seq data next week. So we'll start to think about downloading some data and the tools that we need to analyse that data, and why I'm recommending the tools that I recommend as well. So I'll give some bit of an introduction to RNA-Seq analysis and why we tackle the analysis in the way that we do. Okay, so I hope to see lots of you next week. If you're following along behind, if you're not live, if you're kind of watching on catch up, so to speak, then I hope that everything makes sense. As I've said before, if you are stuck, profoundly stuck, then drop me a line via any one of the number of public forums that I'm available on, and I will do my best to help you. I think for most people who've contacted me that way, I've tried to get back to them and help them as much as I can. I think that there may be still some people who might be stuck on one or two things. So, but do contact me and ask me, and I will do my best to help you. I do promise. My email inbox is crazy at the best of times, but I really am trying to stay on top of my correspondence with this because I want as many people as possible to get the most out of it. Okay, right. I'm signing off now. So I will see you all next week, I hope. And have a safe and restful weekend, and see you next week. Happy Easter, everybody. Ta-ra, bye.